And I will, rather than give an introduction of their backgrounds, I have asked them to please give a little background on how they got from their advanced degree to where they are at Janssen. Thank you. Dr. Copley.
Yeah, great, thank you. So thank you. It's a pleasure to be here today. It's been a while since I was in Ann Arbor, so it's nice to come back. So I'm going to give you an overview of the oncology program as a whole, and then Mike is going to spend most of the time talking about how we use aspects of bioinformatics and computational biology in developing novel oncology drugs. As part of the background, just to introduce myself, I got my PhD from the University of London, came over to the US and did postdocs at Memorial Sloan Kettering in cancer biology, and then moved to MIT and the Whitehead Genome Center for many years. I spent a lot of time in the Genome Center with Eric Lander and working on the Genome Project. From there, I went to work for an ex-Michigan alum, Francis Collins, at the National Institutes of Health. Worked for Francis for a few years in the early days of the Genome Center, and then moved into industry, biotech, and now pharma. So it's a while ago since I was sitting in your seat, but all of us in the company today have academic backgrounds that led us into basically pharmaceutical and drug development research. And so I think one of the things we're here today is to show you it's actually not quite such a big step, a big change from academic research into what we do. So let me go into big background. So Janssen Pharmaceuticals, you can see the logo on the bottom, is part of Johnson & Johnson. So Johnson & Johnson makes baby powder, all that other stuff. We don't never actually see any of that. Janssen is the pharmaceutical arm of Johnson & Johnson, and it has multiple therapeutic areas: cardiovascular disease, immunology, oncology, neurosciences, virology, as different disease areas. And so we're going to focus today on oncology, which is the single largest research area within the pharmaceutical business. It's a global company. We're scattered, in the U.S., Europe, Asia, basically, and sell our pharmaceutical products globally. So we've had a big growth over the last few years. This is a little bit of sort of corporate advertising. It is going, but we've had a run of good luck or success, if you want to put it that way, over the last few years, and launched a large number of drugs. Our portfolio is really totally, revitalized now, with this. And over the last few years, we have been one of the more productive companies, and we'll show you in essence around the oncology program in more details of that as we go forward. But our oncology vision is a bold one. So what we're trying to do is to change the way cancer drugs are developed. Traditionally, cancer drugs are being developed in patients with late-stage metastatic disease, often by inhibiting signal transduction pathways with small molecules to block signaling from the cell that tells the tumor cell to divide and proliferate, and then with all the somatic genomic alterations leading to the generation of the hallmarks of cancer, where the cell is able to evade the immune system, its ability to repair DNA is inhibited, its ability to go into apoptosis is blocked, is to think about basically trying to move the therapeutic process much earlier into the process before patients have developed metastatic disseminated disease. And to do this by a combination of therapeutics and diagnostics, where we can find patients at high risk and intervene earlier. And we hope by intervening earlier, you'll have tumor cells that will be less prone to the development of drug resistance. Because the major problem we face in oncology development today is that a new compound will I'll give you one example, say the BRAF inhibitor that inhibits signaling through the BRAF gene. If you look at basically images of patients with late-stage melanoma, you'll see systemic disease throughout the patient's body. You give them, and if these patients have an active BRAF gene that drives their disease, you give them a BRAF inhibitor. Within a matter of weeks, that systemic disease clears up and becomes invisible radiologically. The patient looks like they've been completely cured. The problem is there is a residual disease that will gradually emerge over a period of time, and that 9 months to a year later, the patient is back where they were now, except with fully drug-resistant disease. So we have an ability to kill off the majority of the tumor cells through therapeutic intervention, but we are unable to kill all of the cells. Over a period of time, resistance emerges, either through mechanisms where the drug will no longer bind to the target, or the cells will activate alternative pathways to evade, blockade a particular proliferation pathway to get around that blockade. But either way, the cell develops resistance to therapy. And so most of our patients still die of drug-resistant disease. We may provide, some survival benefit. The patients may live for longer for a period of time, but we're not curing patients with therapeutic intervention in cancer. And so at the end of the day, we hesitate to use the word cure because it is such an ambitious goal. But at the end of the day, what we're trying to do is to basically make oncology drugs that do much more than provide a few months of survival benefit to cancer patients, but eventually, hopefully, lead to the cure of some cancers and eventually all cancers. I'll skip over this. This is a relatively large group. It is going so, within the oncology organization, we are integrated in a way that from research and development all the way through to commercialization in an integrated group. So this is different from many other pharma companies who basically organize themselves horizontally. We have an integrated team that goes from research and development, from target discovery to target validation, development of small molecule or large molecule inhibitors, into clinical testing and so on, and through regulatory approval. All this is done as an integrated end-to-end team. Typically, it takes about, recently, the fastest drugs being approved take about 3 years from the time a drug is first tested in humans in the clinic, all the way through to approval. Some of the recent drugs have been done in about 3 years. Typically, the process is a lot longer, 5 to 8 years, but with targeted therapy, it's being quicker. And then we also want to be able to take these compounds out, develop combination therapies, integrate them into existing regimes of therapy in ways that we can affect patients most effectively. Our goal for a while was to be one of the top 5 oncology companies. By 2015, this is actually J&J began its investment in oncology in 2008. And at that time was a very minor player in oncology, and we've had a significant growth over those last few years. So now we're the 4th largest company in oncology today. This is going right here. Only Celgene, Novartis, and Roche are bigger than we are. So we're now a large oncology company with a lot of diverse, opportunities. One of the really attractive things about becoming bigger and more successful is the quality of drugs we get to work on is much better. We can now compete to license really active, exciting compounds. We have bigger internal teams able to develop compounds that are essentially much more interesting, much more active in patients. And so certainly one of the advantages of growth is you basically can work with much more exciting and interesting compounds as you develop that capability. We're based across the globe, but the main sites here, Mike and I are based here in Springhouse in Pennsylvania. This is just north of Philadelphia. We also have clinical development in Raritan in New Jersey, just to the north of the site there. And then our other major research site is in Belgium, in Beerse, a town near Brussels and Antwerp in Belgium. So our major research sites are here in Belgium and in the east coast of the U.S. And then we have, affiliated clinical sites globally, in China and Shanghai, Tokyo, also Beijing, other sites in Europe and the U.S. for, clinical development. So, but basically from a research and development perspective, we're focused on the east coast of the U.S. and in Belgium. So one of the things that Mike will talk about in details around some of the computational biology programs that we're using is rather than attempting to address all of cancer. So if you think of, the oncologists here, people, oncology is somewhere around 200 different diseases. It's not just a single disease, but if you look at the histochemical subtyping of the disease, you can identify about 200 different cancers, all of which are driven by different subsets, different somatic changes, different molecular changes, different immune responses, and so on and so on. The cancer is about 200 different diseases. We've tried to focus on a small number of diseases. And so what you'll see today is a presentation around the biology of prostate cancer, the biology of lung cancer, and the biology of hematologic B-cell malignancies. This is about 1/5 of all cancers, but so rather than trying to develop drugs at every therapeutic area and every opportunity in cancer, we wanted to be able to focus on a smaller number of indications, be able to go biologically much deeper into those indications, and then bring forward multiple different drugs intervening in different points in the development pathway over a long period of time, rather than the traditional approach in pharma is basically following an individual compound and perhaps a backup at a time, and then following that into all different indications. We wanted to basically take an approach of going really deep into some limited number of diseases and becoming really expert in those areas. And this is really driven by some underlying biology and science that we'll come to in a minute. So the areas we're going to talk about today are hematologic malignancies, initially thinking about B-cell malignancies, and drugs that basically work through Imbruvica here. This is the drug here. This is a Ibrutinib tyrosine kinase inhibitor. This works by blocking B-cell receptor signaling. So in essence, basically the drug works by stopping proliferation in B-cell malignancies by blocking the B-cell receptor signaling to signaling intracellular signaling. We have Velcade is a proteasome inhibitor. Daratumumab here at the bottom is a CD38, anti-CD38 molecule. Variety of siltuximab is an anti-IL-6 inhibitor. So variety of different mechanisms you'll see here in B-cell malignancies, expanding into myeloid malignancies. The other major area we'll talk about today is in prostate cancer. Two major drugs here. Zytiga is an androgen and Aragon 509 are both androgen synthesis inhibitors. Prostate cancer is really driven by androgen synthesis and signaling through the androgen pathway. It's not a vastly molecularly subtype disease, and all of the disease progression is related primarily through this androgen pathway. And the current therapies in prostate cancer all work through inhibition of androgen synthesis, often in combination with other targeted interventions. But the androgen pathway is really critical, is the critical pathway for essentially all prostate cancer. Lung cancer it contrasts very much to prostate cancer is that it is a very much more molecularly subtype of disease. Different subsets of lung cancer are driven by mutations in EGFR, KRAS, MET, a variety of other individual molecular subtypes, so it's very different in the sense that it's a highly diverse disease, where prostate cancer is a much more homogeneous disease. Multiple different approaches here to inhibition of particular signal transduction pathways or other approaches, and increasingly a focus on immune-based. And what Mike will tell you about is a lot of immunotherapy and immune-based approaches to these diseases. And then we have emerging programs in colorectal cancer. Colorectal being one of the most well genomically or genetically characterized diseases, but still therapeutically somewhat ineffective. Data based on that has still been somewhat limited therapeutic effect. So we have an early program in colorectal cancer, but we'll spend most of the presentation talking to you about the biology and computational aspects of interpreting the biology, drug response, emergence of drug resistance in hematologic malignancies, in prostate cancer and lung cancer. Just to give you some of the diversity of the types of drugs that we see and the ones we work on. Daratumumab is an anti-CD38. It's a monoclonal antibody. Ibrutinib is a irreversible Bruton's tyrosine kinase inhibitor. It's unusual for small molecules to be irreversibly binding because of the potential toxicities from that irreversible binding, but in this case, the toxicities are being quite manageable. Zytiga is a androgen synthesis inhibitor. Siltuximab is an anti-IL-6 um, a monoclonal antibody that prevents the tumor getting access to IL-6. Velcade, a proteasome inhibitor. 509 is another androgen antagonist, androgen synthesis antagonist. And the FGF program this is a small molecule tyrosine kinase inhibitor. This is a nanomolar pan-FGFR inhibitor. So there are actually 4 FGF receptor inhibitors in the genome, more than 20 different ligands that bind to those 4 receptors. And so this is a drug that actually inhibits the signaling out of the FGF receptor for all 4 of the receptors. And this is an interesting program Mike will tell you about, where we're actually developing a companion diagnostic around mutations in the gene to determine who will receive therapy with an anti-FGF receptor. If you have the wild type sequence, then you don't, then it's telling you that your FGF receptor pathway is not activated and that the cell is not driving its proliferation through the fibroblast growth factor receptor, but it's using other pathways to drive its proliferation. So you can imagine if you then try to inhibit FGFR in a patient who isn't activated, you can have no therapeutic benefit. So this is a nice canonical example of using a diagnostic test for the driver mutations to identify patients who will respond to therapy. And so let me skip over this one in the interest of time. A big part of what we end up having to do, of course, is basically find these predictive biomarkers in our drug development program and then develop a regulatory approvable companion diagnostic test. And so when we refer to a companion diagnostic test, we mean is a particular assay that's been approved by the FDA that actually allows you to predict response to therapy. And today there are only 19 of these that have been approved by the FDA, but they're all against tyrosine kinase inhibitor targets, a small number of TKIs. We also have a lot of interest in one of the things emerging around translational research. One of the things we're really interested in doing is looking at circulating tumor cells so that we can actually monitor the molecular changes going on in tumors throughout their course of disease. It's typically a patient, when presents with cancer, they'll have a diagnostic biopsy taken at the time of diagnosis. That is usually the only physical sample that will be taken from a patient during their course of treatment. They usually will not be resampled unless they're in a major research institution. But typically in the course of treatment, the patient's cancer is only sampled once during their course of disease. And we know, of course, that over time the tumor evolves. The different somatic mutations emerge. They get the different selection pressures imposed by therapy, by disease progression in the patients. And so one of the things we want to be able to do is to be able to look in real time at the molecular changes that are occurring in the tumor and adjust the therapy based on what's there today, as opposed to what was maybe there in the diagnostic block, which may have been taken months or years before, and the tumor will clearly subsequently have evolved. So there's a lot of interest in looking at both circulating tumor cells, also looking at the content of the tumor that may be from cells that have escaped from the tumor, and looking at their RNA and their DNA, particularly DNA, in circulating in blood, in plasma, and being able to differentiate DNA from normal cells from cells that are apoptotic or necrotic, and have released tumor cell DNA into the circulation, being able to measure driver mutations and things like that directly from the plasma. But also, here's an example of looking at tumor cells from the blood, and looking here at this is looking at gene amplification using a fluorescent in situ analysis of circulating tumor cells. Again, we're also involved in developing novel genomic and cellular technologies. This is a microfluidic approach to the separation of circulating tumor cells out of the blood. I won't go into detail on this, but this is some of the other things that we do, in essence, where we could basically take a 7.5 ml blood draw and then isolate a few tens or hundreds of CTCs out of that blood, and then be able to then either analyze those in individual cells or pool them and do next-gen sequencing based approaches to the characterization of the tumors directly from the blood without the requirement of needing to do an invasive procedure for the patient. And then lastly, just to mention the immunotherapy strategy. One of the big changes that's happening in the industry right now is an increased focus on immunotherapy. And this is really being driven by recent data that's emerged over the last 3 or 4 years around being able to inhibit immune checkpoints. So for those of you not familiar with this, an immune checkpoint essentially is a ligand that is expressed on the surface of a tumor cell that actually inhibits the T cell response to the tumor and prevents the T cell from recognizing the cell as foreign. So in essence, what happens in this biology is that the tumor cell essentially is cloaking itself. It's hiding from the immune system and preventing the T cell based response to the tumor. And so what was developed with these drugs, for the CTLA-4 and the PD-1 checkpoints is a means of inhibiting this. So in essence, what happens is that when you inhibit that in a patient who's had a tumor with a prior immune response, that prior immune response can reestablish itself. And what's being found is that in a subset of patients, we can essentially have very, very long-term outcome survival benefit to patients, essentially cure these patients of their disease through the immune response. So there's now numerous data on patients surviving 5 or more years post-therapy who previously had stage 4 melanoma. So a stage 4 melanoma patient, you would expect to have a life expectancy measured in weeks. There's a very late stage melanoma. It's an absolutely lethal disease. But now by basically using this PD-1 blockade, you've been able to reestablish the immune response. And so about 15 to 20% of melanoma patients can now be essentially cured of their disease by these immune-based therapies. And what we're trying to understand is what is happening to those patients who have this really prolonged survival, who should have, statistically died within a matter of weeks or months because of the late stages of the disease, yet they're now surviving treatment disease-free years after therapy. And the hope is to be able to take these long-term immune responses and actually, so now instead of being able just to have it work with a minority of patients, be able to understand the immune system and the biology of the tumor. And one of the there was a paper published, I think it was in Science, Mike, yes, they were sort of underlying the hypothesis is that the mutational status of the tumor. So you can imagine as the tumors develop their somatic mutations through the genomic instability in the tumor, and if those then mutations are then converted into RNA, and translated into protein, that those mutations now create neoantigens. You have novel proteins with mutations, and those are being recognized as neoepitopes for the immune system to recognize. And so the theory that's emerging is this somatic instability is creating the production of more and more neoepitopes on these tumors through the somatic mutation that makes the cells more visible to the immune system. So this has to be borne out, but it's an interesting idea of why some cells are more immunogenic than others in terms of the numbers of mutations they're carrying and which of those are actually then carried, converted into protein, expressed as protein. The other areas we're looking at, one of the areas, another exciting area about immunology is looking at measuring T cell infiltration in the tumor. You find patients who have an active immune response where you can detect T cells within their tumor that are essentially located close to the tumor cells and are essentially killing those tumor cells. Is by developing bispecific antibodies. We can have one of the, instead of having an antibody that only binds to one motif, you have a bispecific antibody. You identify one on the surface of a T cell, one on the surface of the tumor, and you can now start to bring and force T cells and tumors into closer proximity and have an accelerated cell killing. And you can look at multispectral imaging at basically looking at T cell infiltration and finding ways and then measuring ways that we can show that the T cells are being more active within the and then lastly, this is my last slide. We do an awful lot of work of collaboration. This is a very small number of working with a lot of academic institutions, a lot of small biotech companies in developing these different technologies and approaches. Belfer is institute in Boston, at Harvard. MD Anderson, we have collaborations with many other, we actually, today, meeting with people today, we have several collaborations with investigators here in Michigan as well. But there's probably a list of about 20 different academic centers that we collaborate with, for these types of approaches. So I'm going to turn it over now to Mike and have him basically give you a more detailed perspective on some of these programs, specifically referring to the biology and the processes of drug development, in these different programs. All right, here you go. And you want this. Okay. Does that work? Can you hear me? Okay. All right, so I'll start with a brief introduction. So I have a bioinformatics background, but I consider myself a biologist. So my undergrad training is in biology. And after undergrad, I actually had the opportunity to work at DuPont for a group that was actually generating assays to detect foodborne pathogens. And so in that process, I was exposed to some of the research fellows there that were generating computational models of the data that we were generating from these assays. And I actually got to know some of this work quite intimately, and ended up programming some of the software to do some of this detection. And this actually led me to the bioinformatics program at Boston University through the prompting of some of the people I was working with at DuPont. And then from there, I ended up at Pfizer after I received my PhD. And then a few years later, well, several years later, I ended up at Johnson & Johnson within the Janssen group. And so now I lead a group that does computational biology under Nick in the Translational Research Organization. And I'm just going to go through, as Nick mentioned, some of the projects that we're involved with. I'm not going to go through too much of the details of these projects. I'm happy to talk with any of you afterwards or by email later. But I just wanted to give you a flavor of some of the projects we're involved with, and really expose you to some of the things we do in pharma. Because oftentimes, if you don't have these types of interactions, it's hard to understand sometimes what it is that we do exactly and how computational biology or bioinformatics actually fits in. But I'll start with saying that it's a critical piece of what we do. And it really is necessary for many of the facets of drug discovery and also in the translational research project. So as Nick mentioned, we partitioned ourselves into these disease area strongholds. And really this was an effort to focus the group, to focus the entire oncology therapeutic area into very specific segments. So as Nick mentioned, we have a heme DAS, we have lung DAS, and a prostate DAS, and then an emerging colorectal DAS. And each one of these DASs is composed of members from different groups, including discovery, our group, translational research, clinical, commercial, and others. And in addition to that, we have computational biology support for my group embedded within these groups. So these groups drive a lot of the scientific strategy, but also feeding into them are the clinical programs. So as Nick mentioned, some of the programs within the heme DAS, including Ibrutinib and daratumumab, a lot of the discussions around those drugs occurs within these DASs. And so this allows the disease area stronghold to feed back and get a better understanding of what we're learning from these clinical studies. So to speak specifically to the computational biology core functions and how we operate within the TA, we integrate all the DAS strategies, as I mentioned, and our goals are to identify exploratory biomarkers and validate proposed predictive signatures and others that we may come across either in the literature or through our collaborations. We also characterize and propose new targets and drug mechanisms of action and resistance for each of the DASs. And I think most importantly, and why it's important, I think, for the members of my group and also when you're considering what you're going to do in the future after you finish this program, is the biological context around a lot of the work that we do. So it's not just generating tables of genes or the most significant pathway that seems to be enriched. It's actually taking that and boiling it down to something that you can speak with the clinician about. So what is it specifically biologically that all of this data actually means? And so that's why I think it's very important that if you don't have that sort of hybrid background, that you really do seek that out. So it's not just about programming. It's not just about understanding the biology. Actually, the hybrid of the two makes you, very valuable to pharma. In addition to that, we also license and build computational infrastructure, and this is a necessary component, I think, of a lot of the work that we do. Many things you can't get off the shelf. We've seen from academic groups and others. Oftentimes, you're the ones that have to invent this stuff, and ultimately, these things become commercialized, and pharma companies and others end up licensing some of this. But even then, off-the-shelf products aren't always available for the needs that you have. So I just wanted to show this as a diagram, really to ground us, about where computational biology fits into each one of these facets. So, regardless of whether you're in academia or in industry, this same methodology applies, where you start with a hypothesis, you generate from that hypothesis, you set up a study. That study generates data, produces some analysis that you would work on, and then hopefully would generate even additional hypotheses beyond that. And for us, we would diverge onto or we would split off and hopefully generate a drug and a diagnostic to go with it. So where we fit in with each one of these is around hypothesis generation. This is very important, I think, to take public data or our internal data and really help our scientists understand what that data means. And this includes both preclinical and clinical data evaluation. And also from the discovery perspective, can we identify new targets that we should be going after? So it's not just target identification, but also validation of targets that they may bring to us. From there, also setting up the studies themselves. So what makes a good study? Helping our scientists understand, how to sufficiently power the studies. And this goes for both preclinical and the clinical biomarker studies that we're involved with. We also have a clinical biostats group that does a lot of the main clinical study work, but we're responsible for a lot of the biomarker design and setup. And then also generating statistical analysis plans that lay out very clearly what it is that we're going to do with the data when it comes back. On the data side, we're involved with data management, QC of the data, data visualization, as I mentioned, infrastructure development. And then finally, from the analysis side, and this is really the bread and butter of what we do, the statistical analysis, data processing, analysis pipeline development, probably a lot of the things that people in this room do, and also developing tools and algorithms. So I won't belabor this because I think everyone in the room probably has some experience doing these things, but I did want to show, again, where these things fit in. So everything from hypothesis testing, where you really do need a very clear and good grounding in statistics, through machine learning, where you're doing classification and you're trying to build models that predict something. So in this case, we could be trying to predict outcome or predict resistance to our drugs. And then also, very importantly, data mining and visualization. This is very key to help our scientists translate this to something that's interpretable by clinicians and others that may be in the data. But ultimately, all of these different analyses, again, feed into another analysis that includes trying to understand the biological context. So what does it all mean? This can include pathway and network analyses, or functional annotations, or other approaches like that. Some of the partners we have, as I mentioned, clinical stats, non-clinical stats. We have a research and development IT group that's specific for us, and they help get us set up on things like the cloud and other infrastructure-related things. We have a discovery sciences group that spans all the therapeutic areas, and that discovery sciences group is responsible for answering sort of the bigger questions. So what could apply to both immunology and oncology? How can they help us design studies around understanding diseases that may span both, or infrastructure that would actually benefit both groups? And then obviously we rely heavily on external vendors, and then also partners within other therapeutic areas like immunology and neuroscience. So to quickly jump into some of the projects that we deal with across the different DASs. So this is the heme DAS sort of overarching picture of what it is that the focus really is. As Nick mentioned, we really want to hone in on specific diseases. And in this case, because heme represents a large number of diseases, we've really tried to focus on specific diseases. So AML, MDS, diffuse large B-cell lymphoma, multiple myeloma, and also other B-cell malignancies that can be targeted with
drugs like Ibrutinib. And just to show how all these diseases are related, if this isn't familiar to everyone in the room, I'll just quickly walk through. So this shows B-cell development and where specific mutations can occur in these B cells and as they travel or as they mature, and where specific mutations can lead to different diseases. So CLL to mantle cell to follicular lymphoma, Burkitt's lymphoma and Hodgkin's lymphoma, and then DLBCL over here. And why this is important is understanding this progression, understanding some of the mutations that occur can help us develop better drugs. And in this case, if we look at a specific protein called BTK that Nick mentioned, this is Bruton tyrosine kinase. This actually plays a part in this entire B cell maturation. And by blocking it, you can actually impact many of these diseases very well. And so what we've done with Ibrutinib is specifically targeted this, and a lot of the results we're seeing in these clinical studies has been very positive and encouraging. So this actually has led to Ibrutinib's breakthrough status for mantle cell lymphoma and CLL, and hopefully we see as good results in some of the future studies we're dealing with. So for mantle cell lymphoma specifically, it's shown impressive results, and this is actually what led to the breakthrough status. But I think one important thing here is something that the computational biology group can bring to the table, and that's really trying to understand the biology of its activity and the potential mechanisms of resistance. So not every patient is going to benefit from Ibrutinib. So understanding why those patients don't benefit is very important for us to consider combination therapies or also lead on to the next drug. I'll skip over this for the sake of time. So just to mention the studies, the mantle cell lymphoma study. So this is MCL 2001. It's also known as the SPARK study. This was a single agent oral Ibrutinib at 560 milligrams. Subjects with mantle cell lymphoma had to receive 1 to 5 prior lines of therapy, previously treated with rituximab or Velcade. And the primary endpoint in this case is objective response rate. So for those not familiar, this is looking at those complete responders and those partial responders to Ibrutinib. So those that seem to have their disease completely eradicated to those that showed some tumor regression. This is what's factored into the overall response rate. So in this study, we saw a 62.7% overall response rate. So very good, but not every patient responded. And so what we did in the translational group is then asked, of the patients that didn't respond, there were 25 who seemed to have progressive disease after the first evaluation. And of those, 23 had DNA that we could extract successfully from their samples. And so from there, what we've done is a targeted panel of 97 genes that are specifically around the BCR pathway, BTK, and some other related pathways that we think would generate some primary resistance to Ibrutinib. And so we've done a deep sequencing of those to about 1,000x, and in addition to that, have done an exome analysis on these samples. So I can't show you all the results from this, but I think some of the results look encouraging, and I think we have some interesting targets to follow up on. But there's a lot more work there, and obviously if you've done this type of computational work before, you know there are many different ways to slice and dice next generation sequencing data. Many different methodologies can be applied to it. Embedding this methodology is something that our group is involved with as well. On the multiple myeloma side, daratumumab, Nick mentioned, and this is about 10 to 15% of all hematological cancers. It's most prevalent. It's the most prevalent heme malignancy in people over 65 years old. And really, there's a huge need. I think that it's a huge market, and the potential for newer drugs with newer mechanisms of action are certainly there. So one drug, daratumumab, is one that we've been taking through the clinic. And again, this received breakthrough status for multiple myeloma treatment. It targets CD38. This is highly expressed on multiple myeloma cells and other heme malignancies. Interestingly, it has multiple mechanisms of action. So everything from CDC, ADCC, ADCP, and MDSC are mechanisms that daratumumab could be using. So one thing that we need to do and really understand is tease out the specific mechanisms of action that are occurring in the patients that respond. So the key translational objectives for this program are really contribute to the dose selection, and then from our perspective in translational research, explore the biomarkers that are predictive of response and ultimately lead to a development of a co-diagnostic. And then there are a couple others that don't necessarily involve the computational group, but are listed here. Okay, so what are we doing bioinformatically? So one of the powerful things about running multiple studies with the same drug in disease populations that are similar, maybe not identical, but similar, is that you can now start looking at cross studies. So here we have 4 multiple myeloma studies that we have ongoing, where data is now coming in or have already had their databases closed out, that we can start mining through this data and get a better understanding of what it is that really is the mechanism in play here. So some of the things that we're measuring is looking at the mechanism of action, ADCC and CDC, and response mechanisms. The biomarkers measured include cell levels and immunophenotyping in PBMCs and bone marrow. So looking at NK cells, activated NK cells, T cells, CD38 expression itself, complement inhibitory proteins, and then also looking at exploratory proteomics panels. So this is looking at somologic panels where you can measure in serum, up to 1,000 proteins. And then in addition to that, looking at SNPs and copy number variation in PBMCs. So taken together, I think this is a very interesting and powerful way to maybe glean some information from these studies that have captured data in a consistent way across multiple studies. Another important thing is minimal residual disease. And this applies not just to multiple myeloma, but also AML and others. And the idea is that if we don't know, we don't really know if the patient's cured just by some test, right? So this diagram at the bottom shows a patient that may be driven into cytological remission and then further into CR. And this would be minimal residual disease positive. In other words, they still have some minimal residual disease. And what is it that we can do with daratumumab and other drugs to drive them even further to a deeper clinical response? So one obvious problem with this is that we lack a current standard for measuring what MRD actually is. And so in this case, there are 3 different assays listed here. There are others. But among these assays, which is the best, which is the most sensitive, which is the most reliable? And this is something that's very important for us to figure out as a community, not just J&J. Because those patients that achieve this very deep remission may in fact go on to generate these long survival tails. But you may also have monitoring of these patients as well over time. So after they are driven into remission, periodic sampling of these patients using the most sensitive assay and most reliable assay you can get your hands on is important to see those patients that would have this bump that occurs later, so you can then treat them with either the same drug or some other drug before they actually have a full recurrence. So early detection of this recurrence is very important. So across all the studies, and I think this doesn't just apply to multiple myeloma, but understanding pre-malignancy or clinically indolent disease and its progression into a more aggressive disease and a disease that's very hard to treat is something that's very important to us as well. So if we look at the disease spectrum of multiple myeloma, and this again could apply to AML and other diseases, where you may start with this smoldering pre-malignant state and ultimately get newly diagnosed multiple myeloma and then relapse refractory disease. If we capture DNA and RNA sequencing data, tumor and immune cell profiling, and also proteomic profiling, we can start to understand and tease apart what is actually happening between the different stages of disease. What makes a patient high risk to move from smoldering multiple myeloma state to a newly diagnosed multiple myeloma patient. And this is something that really can feed back into our DAS pipeline and help us, with new targets for T-cell redirection, chimeric antigens, immune checkpoints, and other targeted therapies. So an interesting project that we're also involved with Heme is a collaboration with the MMRF. And the MMRF is the Multiple Myeloma Research Foundation. This was a charitable organization founded by a patient, actually, that had multiple myeloma. But she also was a pharma executive. And so she had a good understanding of what it takes to actually create new or develop new drugs to treat disease. And she started this as a consortium of 16 leading academic centers. And as part of this Multiple Myeloma Research Foundation is this CoMMpass study. And so this is the goal is to profile and fully characterize 1,000 multiple myeloma patients and then follow them longitudinally for 10 years. So this is a massive amount of data being collected and a huge resource for the community that's studying multiple myeloma. And the goal of this is really to make all of this freely available. So J&J has been a sponsor of this, and by paying into this, we get 6-month early access to the data, but after that, it becomes publicly available. So everything from flow cytometry, RNA-seq, whole exome, and whole genome chromosome analysis. And then also another contribution that J&J has made is looking at circulating multiple myeloma cells. So this plays into the work that Nick had presented with the Veridex platform. So I think maybe I'll just comment here about the different standard of care agents being used. So they generally fall into different classes, so proteasome inhibitors, alkylating agents, and then IMiDs. And it's up to the physician to treat these patients however they want. So what's being tracked among these 1,000 patients is all the different treatments they receive, so you can start teasing that apart as well when you look at the clinical information and compare it to some of the genomics data. So this is a little dated at this point, but as of last year, there were 1,000 patients that had been screened, and 669 of them had been enrolled in this study. So it's accruing fairly rapidly. They're now far beyond that this year with the interim analysis 7 data release, but as of last year, the interim analysis 6. And if you were to go on their website, you should be able to see this data. You'll see about 600 patients are enrolled. I'll skip over that. Just to give a quick idea of the type of data that is being generated here. So this is survival plots broken out by the first-line therapy that these patients receive. So you can see that the confidence intervals really are largely overlapping for many of these. But I think we're getting to a point with the latest data release where we can start seeing separation of these curves. And so this can really help us understand, what are the best combinations? What are the drugs that seem to be most effective? And what are the different genomics that correspond to these types of responses? So do we see different mutations or different things popping up in the genomics data that correlate with better response? And maybe we should be stratifying patients using those different markers. So the path forward for this is to further analyze the data in conjunction with TGen, who's running a lot of the bioinformatics for this, and also with the MMRF itself. Clinical endpoint correlations with the molecular readouts, like I mentioned, will help get a better understanding of what markers are truly or possibly predictive, and then look in a validation cohort to see if those same things can apply. With the lung DAS, I'll just quickly go through this for lung and prostate. I think in general, we're looking for new driver segments of squamous cell carcinoma, acquired and intrinsic resistance to adenocarcinoma, and also a very key focus on small cell lung cancer as well. And so some of the work that's going on within the computational group is looking at some of the clinical data that's coming in from the FGFR studies. And here it's looking at some of the predictive markers that are being proposed. And these are some of the related to some of the fusions, but also we had run a broad panel, foundation medicine panel as well. So there are a few hundred other markers that are in there. Also, pharmacodynamic markers have been tested in the previous clinical study, as well as skin and tumor IHC markers. And so all of this data has to be crunched within our group as well. In addition to the clinical project, we also have a project going on called the Pre-Cancer Genome Atlas. So this is a play on the TCGA. This is PCGA. And this is a project going on with a collaborator at Boston University. And this is, the goal of this is to look at 26 high-risk COPD patients, followed longitudinally with sequential airway brushes and lesion sampling, and then also perform RNA-seq and exome-seq on those samples to get a better understanding of what's actually changing during the progression of disease. So related to this, in a similar vein at least, is a project run in China. This is the China Choice study, and this is being run with our J&J China collaborators. So the goal here is 250 patients, 7 sites, targeting 125 adenocarcinomas and 125 squamous cell carcinomas, and then also run RNA-seq and DNA-seq on the same subject. In addition to this, we have a couple of discovery projects going on. Synthetic lethality is something that was proposed and we're moving forward with. This is looking for identification of loss of function alterations that can be used as classifiers for a functional paralog. And so I'll go into that in a minute. And then also looking at signal peptides, where we can look in gene expression databases to see if we can identify signal peptides that have a particular domain, which show differential expression between lung tumor versus normal. So just to jump into the goals of the PCGA project I mentioned, the key questions here really are define the spectrum of genomic alterations that are changed from pre-malignant, identify the molecular alterations and pathways associated with lesion progression, identify new candidate targets, and this obviously can feed into our discovery group, and then refine and validate these genomic alterations associated with lesion progression using prospectively collected samples. And then finally, perform functional assays. So this would be the follow-on to some of the discoveries that we might make with this data. The China Choice study, the key questions really fall into different categories here for gene expression or somatic alteration. So for gene expression, we can start looking for genes that are show unique expression profiles or things that differentiate smokers and non-smokers. And then for somatic alterations, really understanding is there a difference between Eastern and Western populations? We know from TCGA what to expect, but that may not necessarily be what we find in this China study. And also, can we develop classifiers as we would for any of the clinical studies we're running? And then finally, are there novel recurrent fusion events, or can we identify key resistance markers from this data? Synthetic lethality, as I mentioned, this is where the functional inhibition of one target is efficacious exclusively in cells with a second functional deficiency. So the diagram here just illustrates if you have a wild type cell, if you knock out one of the two partners, you see no effect on viability. However, if you knock out a gene in a cell that already has a mutant in a different gene, you actually can induce cell death. And so this is the concept behind synthetic lethality. This is not a new concept, but we're looking at this in the context of some of the new data sets that have come out. So trying to leverage some of the public data, TCGA and others, where we can start looking specifically at this using both gene expression and the somatic mutation data from the DNA-seq experiments that have been run, and see if we can tease out some of this information and perhaps propose new targets for lung cancer. So for the prostate test, I just have a few slides, but as Nick mentioned, I think a lot of the clinical focus right now is around the AR axis of prostate cancer, so targeting androgen receptor signaling. And so here we're looking for computation. We're looking to identify those patients that may be at high risk. Right? So a lot of patients that have prostate cancer won't necessarily die from prostate cancer. So a lot of the work here is really trying to identify those patients using molecular signatures or other assays that can help us distinguish those patients that would be high risk from those patients with indolent disease. So we're trying to really focus on that using computational methods within my lab. So just to go into this a little bit, as I said, understanding key resistance mechanisms is also important, as it is in heme. So if we can start to tease them out from the clinical data we're generating, that would certainly help with the next round of drug development that we're doing, and also try and identify these high-risk markers and pull up that population and try and treat them more aggressively. The last thing on the list here is characterizing IO and checkpoint gene expression. So just to highlight that here, looking at expression profiling of immune cell populations in tumors and peripheral blood really can help us understand the IO makeup of a particular tumor, and perhaps understand better what's going to be predictive of patients that are more likely to respond to our drugs, or patients that would benefit from some combination with an IO therapy. So I'll jump into some of the cross-staff projects, and then I'll end with this. So obviously, we're working across many different disease areas, but there is a common thread in the types of computational infrastructure that we need to do this work. So we'd like to leverage both external and internal data and analytical methodologies to deliver on the DAS objectives, effectively communicate within and across DASs, store data in a compliant way, and then standardize reproducible research. So one of the things that we've built is this NGS pipeline. And this is actually leveraging the Amazon cloud. I know when you're here, you probably have access to large clusters, and somebody maintains that for you. In industry, we have the same infrastructure, except it's very expensive to maintain, it's very expensive to upgrade, and over time, it's very costly. So something that's been commoditized is this high throughput type analysis that you can farm out to Amazon or other cloud providers. And this is something we've embraced and really driven down the cost of doing this type of research. So this just illustrates the system we've set up to take advantage of this really infinitely scalable system for doing data analysis. And then internally, we've built this Omics data capture system. And really where this becomes important is when people have questions about previous clinical studies or previous preclinical studies, right? Oftentimes, the data in the past would be squirreled away on someone's hard drive somewhere. Even in academia, things are very siloed depending on what lab you might be in. So this is an effort to really harmonize how we're storing data, harmonize how we're querying that data, and make it accessible not just to bench scientists who have questions, but also to the computational biologists who might be able to run more advanced queries on it. So in addition to a Spotfire interface, where we have a very nice point-and-click style way to generate plots and things like that and filter data, we also have an API that allows programmatic access, and that actually accommodates the people in my group who really want to dig into the data and maybe slice and dice it in many different ways. So this is just an example of a heme cell line profiling project that we have in here, where we have gene expression profiling data by microarray, DNA-seq, RNA-seq, and flow cytometry data all combined. And this allows very quick manipulation of this data and filtering so that you can quickly get to answers when in the past it would have been much more difficult to do. And finally, I'll end on this. This is some of the efforts within our organization, really company-wide, is this idea of reproducible research or discovery data integrity, this DDI. And this is really important, not just for us, but I think also to the academic community. If you read some of the papers that show how irreproducible a lot of the data analysis and really studies have been that have been published, I think this really gets at the heart of that, really having analyses that you could hand the script to anyone and they could rerun it. And this is a concept that I think has been embraced by some labs, but not all. But I think it really gets at the heart of the whole idea of this discovery data integrity, that for every plot that we generate, for every report that we write, we can actually trace it back to the original raw data and the script that generated it. So for a lot of the work we do is in R, and this is really a nice fit for something like this. Everything is scripted, and we know exactly where all the data is for these. So just to summarize, the DAS alignment really keeps projects focused, and I think it keeps focus within the groups. Many clinical studies incorporate omics or high-dimensional data readouts, and so this is all work that comes to the computational biology group to process and analyze and help interpret. Disease interception is a key strategy of the DASs, and immuno-oncology will certainly play an important role in cancer treatment going forward. So understanding what is it, or what do we need to be looking for in these assay results to tease out to really inform how to best treat patients is something that we have a really important thing to keep in mind as we're doing this. Computational challenges, biomarker development. You know, hear a lot about big data. You may see Johnson & Johnson and other pharma companies and think, you know, we're sitting on this mountain of data, and in some ways we are. But in many cases, the big data is really the assays themselves. The data sets themselves tend to be fairly small for some of our phase 1 and phase 2 studies. So the question really is, how do we glean as much information as we can from these studies, knowing that in some cases they're often underpowered to actually detect some signal, and then from there generate a hypothesis that we can then prospectively test. Another question that comes up is global versus targeted analyses. So, you know, do you sacrifice depth for breadth or vice versa? So when you're talking about NGS approaches, for example, do you go with a targeted panel where you can get 1000x coverage, or do you do genome-wide or exome-wide and you know, risk missing things that may be showing up at a very low level? And again, are the studies sufficiently powered? With immuno-oncology, some of the computational challenges are really the novel analytical methods that are needed. Some of these have very specific readouts and very specific analytical approaches. And I liken the work that's going on in immuno-oncology right now to some of the early days of microarray development, where it's a bit of the Wild West, where you have many academic groups publishing different methods for analyzing this data. But in reality, we don't really have a consistent way to look and query this data statistically. For tumor microenvironment, the sample heterogeneity can really confound analyses, and for disease interception and minimal residual disease, definitions of pre-malignancy and MRD are not really yet standardized. And so we need to get to, as a community, we need to get to a point where these things really are standardized, and we really do have standard methodologies that are applied. And I think that will help us on the computational side when we're analyzing this data. So with that, I'll just put the acknowledgements up and highlight the 3 people in my group, the computational biology group listed here: Tina Kasnaoff, Mike Gormley, and Chris Moy. And then also the leads of each one of the DASs within our translational research group are underlined here. So Kate Sasser leads the heme DAS, Deborah Ritchie leads the prostate DAS, and Suso Plotero leads the lung DAS. And so they're all responsible for the projects that you saw here. So with that, thanks. Any questions? Quiet bunch.
So you are saying that you Janssen use R to do most of the analytical analysis, but my question is, I've heard that do you need to do some robust tests when you're using R packages? Because I know that some companies, they require to do that.
Yeah. Yeah, so that's a good question. So typically for the clinical stats group, they use SAS, and the SAS has been robustly tested. Right? With R, a lot of the functionality that's present there is not available anywhere else, right? So if you look at something like Boom-Lemma or something for doing RNA-seq analysis, there is no equivalent in SAS. Right? So the evaluations we do just make sure internally that the version of R we're using is standardized and that we're all using the same version of Boom-Lemma. Right? And so this is something we do internal to our computational biology group. But a lot of the work we do here is exploratory as well. So we don't necessarily have the same rigor that's necessary for dealing with things that will end up in a clinical study report that gets filed with the FDA. So it's a bit different.
What proportion of the current efforts are moving away from, say, computer-aided drug design towards, say, one of these therapeutics that you mentioned?
So what, sorry.
So you described a couple of these projects. So of course, traditional ways, after these initial analysis, then in the development stage, you would look forward, towards drug screening and so forth. But you also mentioned, moving away from the biomarker analysis. I'm talking towards the, the targeted therapeutics and such. So what proportion of these current efforts, or the, as you mentioned, DAS, are looking into these new therapeutics versus just the traditional computer-aided drug design?
Yeah. So well, I think I can answer your question in two ways. So I think this is a question partly about small molecule development versus large molecule development, right? So a lot of the assets that we have coming in are actually large molecules. If you look at something like Ibrutinib, we licensed that in, right? So there was not much refinement to the drug itself when it came into us. Yeah. Do you want to comment on that? Yeah. So we also have these the KRAS D12C compound that was just released, right? So that will be coming online, and I think there will be some of this development as well that may be necessary, but we'll see. So I'd say the requirement is diminishing. I think we're getting away from small molecules, but not completely. Is that fair, Nick?
Change this and ask like a general question for your interns and for postdocs and people who start working in your company.
Yeah.
Any company, there's always been the concern of the freedom to publish the result or the process or delays in publishing. Can you, I missed the first part of the talk, if you address that, but otherwise it would be a great idea to discuss that.
Yeah. Yeah, so I think it's a good question. And I think it's, J&J, I think, really goes out of their way to say, publish as much as you can. There are certain things where there are always going to be restrictions, right? So if you're talking about one of the compounds, it does have to go through some process. But in general, if you're doing other types of research that don't necessarily impact a particular drug or a particular development of a drug, there is broad freedom to publish, and it's actually encouraged. It does help, attract other people to the company that are good. For the clinical studies themselves, obviously we try and get on as many of these publications as possible. If we're doing the work here, obviously we feel entitled to get on these publications as well. So, but yeah, there is some delay in getting on these clinical publications. That's right. And I should mention we do have an open postdoc position now. So if anyone is interested, or if you're finishing soon, or other people that are interested in a postdoc position, I think we do have a project mapped out. We're certainly not fixed to that if someone comes in with specific expertise in something else. But this can give you some idea of the types of projects we would like this candidate to work on. And as Nick mentioned, all of that would be publishable, and it's really a requirement of the postdoc itself.
Now, the problem for 10 minutes is that point we can just like people can talk to you informally.
Okay. All right. Sounds good. Thanks. Thanks for inviting us as well.